Yes, we continue our life science seminar today with Elicera, with the company presentation, and I'll give the word to the CEO of the company, Jamal El Moussain. Is that the correct pronouncement? Anyway, the word is yours.
Close enough. Jamal El Moussain.
Okay.
Thank you. So I'm the CEO of Elicera Therapeutics, which is a cell and gene therapy company focusing on immuno-oncology. And what we mean with this is that we develop therapies that uses the patient's own immune system to fight cancer. It's based on many years of research performed by Professor Magnus Sand and his research team at Uppsala University in Sweden. And they've been very successful in securing different types of research grants throughout the years, which has enabled them to develop the projects in quite advanced stages in academia. So we turned to investors actually first time last year in an oversubscribed share issue where we took in 13 million Swedish kronor. And we recently concluded an IPO and financed the company with 62 million Swedish kronor before issue costs, which lasts us until mid-2023. So that the share is now traded on Nasdaq First North growth market here in Sweden. We have a broad pipeline of different drug candidates. So we're developing 2 CAR T cells and 2 oncolytic viruses. Regarding the CAR T cells, there are other companies developing similar products, only 14 in Europe and only 1 in Sweden, being Elicera. And we have our own special take on these types of products, which I will go through during this presentation. We have a phase 1/2 ongoing clinical study in neuroendocrine tumors with an oncolytic virus. And we also have a fully developed technology platform, which we call iTank, which can be used to optimize CAR T cells. Not only our own CAR T cells, but also the CAR T cells of other companies. So this is a technology that we think should be interesting for any CAR T player. We gene modify our drug candidates so that they elicit a second mode of action by activation of the patient's own killer T cells. So this means that we can offer a multiple mode of actions in one drug candidate, which is what separates us and makes this quite unique. We have relevant experience within the board and management. We have clinical experience from both CAR T cells and oncolytic viruses. Our co-founder and chief scientific officer, Professor Magnus Sand, was actually part of the very first CAR T cell study ever performed in Europe. And this, of course, minimizes the risk that we won't be able to conduct the clinical studies in the way that we've planned and do that efficiently. Myself, I have almost 15 years of experience from running different types of biotech companies, mainly in the field of immuno-oncology. I was CEO and active co-founder of another immuno-oncology company, today listed on Nasdaq small cap, called Immunicum. And we also have a board with broad and complementary competencies, with competencies from business development in life science, regulatory expertise, with a focus on immuno-oncology and cell therapies, CMC. For example, we have one board member that was actually part of bringing Novartis Kymriah, a CAR T cell, to the market. So we're very proud of the composition of the board. This slide shows our product pipeline, and you can see that we have drug candidates in different development stages. The most advanced is ELC-100 in neuroendocrine tumors. It's an oncolytic virus. We're going to treat 12 patients in this first step of the study, and we've so far treated 7 patients, but I will talk a little bit more about this shortly. ELC-201 is our, so to speak, next generation oncolytic virus. And what we mean with this is that we have genetically modified this product to elicit a combination of 3 mode of actions in one drug candidate. It's also a oncolytic virus that is applicable for treatment of most cancers. ELC-301 and 401 are our CAR T cells under development, and 301 is in liquid tumors. Initially, we will target non-Hodgkin's B-cell lymphoma. This is the CAR T cell that is closest also to patient studies. So we're currently producing these CAR T cells according to GMP. And our plan is to initiate the first clinical study by second half of next year. 401, initially we will target glioblastoma multiforme, which is an aggressive type of brain tumor. but we will also target other indications, as we think that the target that we're going for is overexpressed not only in brain tumors, but also in a range of other solid tumors, like colon cancer, pancreas cancer, head and neck cancer, and a couple of others. And ELC001, or the iTank, as I mentioned, is a fully developed technology platform which we use to optimize our own products, but which we also believe can be used to optimize the products of other companies in similar fields. So what is an oncolytic virus? It's a virus that you have genetically modified to selectively infiltrate and replicate only in cancer cells, but not in healthy cells. So when this virus replicates in a cancer cell, it does so until the cancer cell bursts and dies. So you get what is called an oncolysis, a tumor cell death. And this is a process that in itself will activate the patient's immune system. So you will have activation of the patient's own T cells that will attack cancer cells in parallel to the virus particles that continue to attack new tumor cells. So basically, you can say that an oncolytic virus inherently has a combination of two mode of actions. And what differs ELC100 to ELC201, which are our two oncolytic viruses under development, is that ELC100 is restricted to treatment of neuroendocrine tumors, while 201 is applicable against most cancer indications. ELC100 also gives rise to these two mode of actions that I just described here, while 201 we have genetically modified to elicit a combination of three mode of actions in one product. So the ongoing study was initiated in 2016, so a couple of years ago. You can see the patient that was first treated in the picture here to the right, and below there you see Professor Magnus Sand and Vince Hamilton, who is a businessman that donated a large sum of money for us to be able to initiate this clinical study. That's also why we named this product after him, Advince. So the study is conducted in two steps, where step one is a so-called dose escalation study in 12 patients. What we're after here is to investigate the maximum tolerable dose to then be tested in step two in an additional 12 patients. The study is conducted in collaboration with Uppsala University that so far have reported promising efficacy data from one of these patients, which is very encouraging. And going over to CAR T cells. So a CAR T cell is a new type of immunotherapy. So the term immuno-oncology is quite a broad term. You have different types of strategies to use the immune system to fight cancer. And CAR T cell therapy is one, you can say, new and very exciting type of therapy within immuno-oncology. ASCO, which is one of the world's largest cancer organizations, named CAR T therapy Advance of the year 2018. And the reason they did this was because CAR T cell therapy turned out to be able to basically cure patients with different types of liquid tumors that previously were very difficult to treat and that had already undergone standard treatments and that were more or less dying and had no other alternatives. So with CAR T cells, you could more or less eradicate all tumor cells. So what this is that you take out blood from the patient, you filter out T cells, and then you genetically modify these T cells so that they express a CAR molecule, a chimeric antigen receptor. And this CAR molecule then recognizes a specific target on the tumor cell. So you expand these CAR T cells in test tube and you re-inject them into the patient. And the idea is then that they will seek out all the tumor cells that carry these targets, bind into them, and then start killing them. And as I mentioned, this works very well in liquid tumors, but unfortunately, it doesn't work so well in solid tumors. And the two main reasons for this is that a solid tumor has a very hostile tumor microenvironment that suppresses the immune system. So it's very difficult for these CAR T cells to survive in this hostile microenvironment in solid tumors. And you don't have the same situation in liquid tumors. And also, there are very good targets for liquid tumors. You have, most often you use CD19. Sometimes you can also use CD20. Which are two very good targets on, for example, B-cell lymphoma. But in solid tumors, it's very difficult to find a good target that is also not expressed on healthy cells. So you basically, all tumor cells cannot be attacked by CAR T cells in a solid tumor situation, because there's a lack of relevant antigens. And also, when the patients relapse, they often also lose the target for the CAR T cell, which means that you can't continue to treat them with the CAR T cell because they won't have anything to bind into. So basically, CAR T cells need to be able to survive in the hostile tumor microenvironment and also trigger an immune response that can attack CAR antigen negative tumor cells. And this is precisely what we believe that we can do with our CAR T cells under development, thanks to the iTank platform. And this is also something that we believe we can help other companies developing CAR T cells to overcome these two challenges. To also further put this into perspective, you can say that CAR T cells have been developed in different generations. So first generation didn't function adequately for different reasons. So due to that, you improved the CAR T cell in second generation, and then further improved it into third, and then finally fourth generation CAR T cell. And what differentiates the fourth generation CAR T cells from other generations is that fourth generation CAR T cells include a cytokine transgene. A cytokine is like an immune stimulating substance. So you have this gene that codes for an immune stimulating substance to trigger a parallel immune response by the patient's own T cells. And when fourth generation CAR T cells uses a cytokine transgene, they only use one substance, one gene for one substance, basically. And what differentiates Elicera's fourth generation from other fourth generation is that we, instead of having a cytokine that we code for NAP, which is a neutrophil activating protein from Helicobacter pylori. And when NAP is activated, it actually releases a whole set of different cytokines and chemokines, up to over 20 of these, which we believe gives a more optimal and broad immune response against cancer. You can also see here that most CAR T cells under development are still second generation, as many as 70%. And only 16% belong to fourth generation CAR T cells. And of these, most of them are actually being developed in academia. So we have only identified one other company that develops fourth generation CAR T cells, and it's a Japanese company called Noile Immune Biotech. And if you look at their news flow the last couple of years, you see that they have been able to successfully generate multiple licensing deals and collaboration agreements with other CAR T companies, both big pharma and larger biotech companies, around their Prime T technology, which is very similar to iTank, but where we believe that iTank again gives a more broad immune response against cancer. So I won't go into the mechanism of action of the iTank platform, but just to conclude the main advantage here of this technology, which is that we elicit two mode of actions. So normally a CAR T cell functions on its own. It targets the target on the tumor cell, binds into it, and then kills the tumor cell. And usually you talk about one target, maybe sometimes two. But the tumor cell, of course, contains a whole set of different targets, tumor antigens, many different ones, not only one or two. There's several that are relevant. And the CAR T cell can only target very few of these. So what we are after is to activate the patient's CD8 positive T cells, which are the killer T cells, against a whole set of different relevant targets on the tumor cell. That's why we have different colors here on point number 2 of the targets, to exemplify that we have a more broader attack on cancer cell. So I've written here that there is a large interest in CAR T cells, but it's still a relatively young field. And what we mean with this is that most of the CAR T cells under development today are actually no later than in phase 1/2 development, as many as 95%. So only 5% of the CAR T cells under development are in phase 2 or later. So there's still a very good opportunity here to make an impact in this field for Elicera. There's roughly over 100 CAR T companies throughout the world, 50-50 split more or less between US and China, a little bit more companies in China, very few in Europe, and only one in Sweden, being Elicera. And most of the CAR T cells under development target CD19, which is this target that is overexpressed on different types of liquid tumors. There are 5 approved CAR T cells today, but only in liquid tumors. So none is approved yet in solid tumors, which is due to the challenges that I described before, and the challenges that we think that we can meet with our iTank platform. So we target in our first clinical study here CD20 and not CD19 in B-cell lymphoma. And this is an indication that actually already has 3 approved CAR T cells for treatment of this indication. And I've told you that it works very well in non-solid tumors. So you can ask yourselves, why would you target these patients if they already have 3 approved CAR T cells and they function so well? Well, it's because recent studies have shown that 40% of patients have an ongoing complete response. And 40% of patients with an ongoing complete response is, of course, amazing results. It's very good data. But this also means that the majority of patients are still in need of additional CAR T treatments or other treatment alternatives, even though you have these CAR T cells approved. And the 3 approved CAR T cells in this indication all target CD19. So we go for CD20, which is a different target. So you can give this either before or after treatment with CD19 conventional CAR T cells. And again, as you remember, we optimize our CAR T cells with the ITANK platform, which means that we will also generate the patient's own T cells to target other antigens on the tumor cells besides CD20 and CD19. So we currently have an ongoing GMP production of these CAR T cells, and we expect to initiate our first clinical study second half of next year. Our business model is based around out-licensing, so we don't have an ambition to take any product all the way to the market. Rather, we want to minimize risk and create value in the portfolio by establishing proof of concept in clinical studies before out-licensing. And there's multiple opportunities. If we stick to the CAR T side here, we see that the ITANK platform is, of course, the basis of both our CAR T cells under development. But as I mentioned before, this is a fully developed technology platform, which we believe can be out-licensed on its own, more or less already starting from now. So this is something that we will start working on after summer here to start also marketing Elicera towards the industry. Since Elicera is still a relatively newly launched company in that sense. So I expect there to be a growing interest in this platform. 301 is a little bit more niched, as I mentioned, in a few non-solid tumors, while 401, we see a broader potential for out-licensing in many, not all, but many solid tumor indications. It's a little bit the same situation for oncolytic viruses, where ELC100 is very niched against the neuroendocrine tumors, but where we have multiple opportunities in 201, being that we can treat most cancer indications with this oncolytic virus. The clinical development plan for the coming 3 years you see in this slide. So we recently financed the company with 62 million Swedish kronor before issue costs. It should last us at least until mid-2023. We expect to conclude the first step of the ongoing phase 1/2 study by second half of 2022. We also expect to be able to initiate our first CAR T cell study by second half of 2022. And then in 2023 and onwards, we expect to have a quite broad clinical program. And we also believe that the company, the size of Elicera, can manage to conduct this quite extensive clinical program in the phase 1/2 phase. But going into phase 2 studies, then we will eventually need to start prioritizing between the projects. And that prioritization will depend on the data that we've collected and also, of course, partner interest. And already starting from the fall, we will start to market ourselves and the ITANK platform to the industry with the hope of being able to generate licensing agreements for that technology platform also. So a short summary. It's a newly launched immuno-oncology company focusing on cell and gene therapy. Substantial research grants have been secured throughout the years. So we have over 100 million Swedish kronor that has entered the company so far. We are recently listed on Nasdaq First North Growth Market here in Sweden, where the share is traded. We have a broad product portfolio with CAR T cells against both solid tumors and liquid tumors. Next generation oncolytic virus under development, ongoing phase 1/2 clinical study. We have a unique positioning with our 4th generation CAR T cells. As I mentioned, very few companies are doing this. Most of them are 2nd generation. We're also very unique with our next generation oncolytic virus with the combination of 3 mode of actions. And from a geographical perspective, very few companies in Europe developing these types of products, and here in Scandinavia, even fewer, of course. And we have a team with relevant experience, clinical experience from both CAR T cells and oncolytic viruses, management team with experience from immuno-oncology, and board with complementary and relevant experiences from immuno-oncology, life science in general, and cell therapy in specific. So thank you for that, and now I would like to open up for any questions you might have.
Yes, but unfortunately, the time is out, Jamil. I'm sorry to say that, but you had your 20 minutes, so I don't think that there will be enough time for question at this, but we will have to do a reschedule and have another event with you where we can welcome questions. So thanks for the in-depth presentation. No, it was a good and in-depth presentation that provided a lot of value to us also. I think that you answered many of the questions that many of might have, but let's look into an additional event for you guys.
Okay, thank you.
Thank you.